Hello students, today I Swati Verma, Assistant Professor at School of Medical and Allied Sciences, Calcutta University, shall be discussing with you about cloning vectors. So if we talk about what are vectors, then a vector is a DNA molecule which is used for transporting exogenous DNA into the host cell. So basically a vector is your DNA molecule that is used for transporting your exogenous DNA, that is external DNA, into the host cell. Now the general characteristics of a vector. What should be the basic characteristics of a vector? So it should have an horizon of replication. The vector should have an horizon of replication that is known as ORI, so that the vector is capable of autonomous replication inside the host organism. It must possess a compatible restriction site for insertion of DNA molecule. A vector should always harbor a selectable marker to screen the recombinant organisms. This selectable marker can be an antibiotic resistant gene. So a vector should always have a selectable marker so that it can screen out the recombinant organism. This selectable marker can be an antibiotic resistant gene. For easy incorporation into the host machinery, a vector should itself be small in size and be able to integrate large size of the interest. So now if we talk about cloning, what is cloning? Then your cloning is a biotechnology which refers to the process of creating clones of organisms or copies of cells or DNA fragments. So it is basically a process of creating clones of organisms. In this, we create clones of organisms or copies of cells or DNA fragments. A cloning vector is also a fragment of DNA which is capable of self-replication. So it is a fragment of DNA which is having the capability of self-replication and stable maintenance inside the host organism. It can be extracted from a virus, plasmids, or cells of higher organism. So we can extract out a cloning vector from the body of virus, plasmid, or cells of a higher organism. The cloning vector must possess the following general characteristics. So here I have enlisted certain general characteristics that your cloning vector must exhibit. That is, the first is it should be small in size. The size of the vector should be small. It must have an horizon of replication. It must also be compatible with the host organism. It must possess a restriction site. So these are your four characteristics that a cloning vector must possess. Next comes to your cloning vector. Now your cloning vector used as the vehicle of transporting foreign genetic material into another cell. So they are means of this, so they will act as a vehicle of transporting your foreign genetic material into the body of another cell. This foreign segment of DNA is replicated and expressed using the machinery of the host organism. So now that foreign DNA that we have transferred is replicated and expressed using the machinery of host organism so that it can express itself. Now this is your DNA fragment. This is your vector. By means of ligation, we will join both the DNA fragment to the vector and we get a recombinant DNA. So now your cloning vector facilitates amplification of a single copy of DNA molecule into many copies. So your cloning vector works as your amplifier. So it will amplify your single copy of DNA molecule into many copy. Now we have different types of your cloning vectors like your plasmids, bacteriophages, phasmids, cosmids, artificial chromosomes, bags, yaks, hacks. These are your retrovirals. These are your different types of cloning factors. Now if we talk about your plasmids, so these were the first vector to be used in gene cloning. They are naturally occurring and autonomous replicating extrachromosomal double standard circular DNA molecule. However, not all plasmids are circular in origin. They are present in bacteria, archaea and eukaryotes. So plasmids are present in bacteria, archaea and eukaryotes. The size of plasmids ranges from 1 kb to your 250 kb. Now the DNA insert of up to 10 kb can be cloned in the plasmids. So a size is your 10 kb. So 10 kb size of DNA insert can be cloned into the plasmids. The plasmids have high copy number, which is useful for production of greater yield of recombinant plasmids for subsequent experiment. The low copy number plasmids are exploited under certain conditions, like the clone genes products produces the protein which is toxic to the cells. Plasmids only encode those proteins which are essential for their own replication. So plasmids will encode only those proteins which are essential for their own replication. The protein is located near the ORI, that is your horizon of replication. Now if we talk about the nomenclature of plasmids, so the nomenclature of plasmids is this is the structure of your PBR322 plasmids. This is your amplification region. Here we have your EcoRI1 in third tet R say 1. So your PBR322 cloning vector has the following elements. It has a plasmid represented by your P, B your Bolivia, that is your name of scientist, R is your Rodriguez, that is name of the scientist. So this is your PBR. P refers to the plasmid, P is the name of the scientist, R is the name of scientist, that is Bolivia and your Rodriguez. Now your 322 number refers to the number of plasmids discovered in the same lab. So your most commonly used cloning vector is your PBR322. It has unique restricted sites PVUL and PSTL ampicillin, BAMHI and SALL tetracycline, BAMHI gene encoding your resistance to tetracycline. Now your PBR322 has 4361 base pairs, so it has 4361 base pairs. It has an horizon of replication, that is your ORI. This is your horizon of replication. Antibiotic resistant genes are your AMP and TET. So your AMP and your TET are your antibiotic resistant genes. ROP gene regulates replication for 20 copies of the plasmid per cell. Now if we talk about your bacteriophages, this is the structure of your bacteriophage. So bacteriophages or phages are viruses which infect bacterial cells. So these are your viruses that will infect your bacterial cell. The most commonly bacteriophages utilized in gene cloning are phase lambda and M13 phase. A maximum of 53 kb DNA can be packed into the phase, so the maximum size is your 53 kb. If the vector DNA is too small, it cannot be packaged properly into the phase. So if size is very small, then it cannot be packaged properly. The examples of bacteriophages are your phase lambda, M13 phase, etc. Now we talk about your plasmids. So these are preferred artificially. This is the structure of your plasmid. It has a promoter area. This is your coat protein. This is your secretion signal, and this is your displayed peptide. So plasmid contains the F1 horizon of replication from F1 phase. They are generally used as a cloning factor in combination with M13 phase. It replicates as a plasmid and gets packaged in the form of single stranded DNA in viral proteins. Now if we talk about cosmids, what are cosmids? So cosmids are plasmids. They are capable of incorporating the bacteriophage lambda DNA segment. This DNA segment contains cohesive terminal sites, that is your cos sites. Cos sites are necessary for efficient packaging of DNA into lambda phage particles. Large DNA fragments of size varying from 25 to 45 kb can be cloned. They are also packaged into lambda. This permits the foreign DNA fragment or genes to be introduced into the host organism by the mechanism of transduction. So they can also be packaged into lambda. Now their packaging will permit the foreign DNA fragment or genes to be introduced into the host organism by the mechanism of transduction. Thank you. For more information, you can mail me on this particular mail ID.